高老师 ，Can you hear me？ Hello, can you hear me?
刚才没听到。重复一遍，现在再测试一下，我的声音能听到听不到？
Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment&Health. Eco-Environment&Health is a journal initiated by prestigious scientists with a strong editorial board, including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design, and broadcasting. The experts will accompany you like a harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision-making that deeply integrates informatics, ecology, and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed of the manuscript status throughout the entire process. The 14-day rapid peer review will make the publication of your research one step ahead. Your distinguished work is worthy of being known worldwide. We will promote your research globally in a three-dimensional manner through various social media platforms. In addition, the open access fee will be waived for the first 3 years, making your research easier to be read and cited by your peers. We continue to explore and innovate the service strategy of the journal. EEH leads ecology and environment to a healthy future.
高老师好，石老师好，
你好，
我是 EEH 编辑部
嗯
唐小燕，
嗯
欢迎石老师。
谢谢您。我刚才是问我所有人听都听见我是您回答的。
是的，是的，我没有，当时我是没有在那个，是在观众席里，还没进来，刚才不好意思，石老师。
没事没事。
可能高老师，那就您等会儿来主持，感谢您的支持，
可以，感谢我们石老师百忙之中抽出时间来给做报告。
Thank you for the invitation.
有机会去武汉看石老师。
请欢迎来，我们高老师很有钱的。
好吧，那非常开心。
高老师是大老板。
不敢。
好的，高老师，那高老师您准备一下，我先退出，到时候你们马上开始主持啊，时间可能快到了。
快到了，差1分钟。
好，感谢石老师和高老师。好，
谢谢你。是不是可以开始了？时间都到了，高老师。
好像没赶我们切换出去，是不是？
已经9点过1分了。
对，我也是感觉。我问他一下，我感觉他是不是他应该按理说给我们切换到一个屏幕上。
现在您主持嘛，然后完了以后我最后就共享屏幕就可以了。
那要不石老师您先共享屏幕是吧？
好的，我可以共享屏幕。
共享屏幕，咱们俩头像也还在。他说可以。
你还得要允许我看看能不能共享。你得要允许我共享屏幕。
他现在不允许您共享屏幕是吧？
还是不允许。
好，那我先介绍吧，然后给他发个消息是吧？Good morning, everyone. Good morning, Professor Shi. It's my pleasure to introduce Professor Shi for this EEH talk. Dr. Liang Shi is a distinguished professor at School of Environmental Studies, China University of Geoscience at Wuhan. He has published about 140 peer-reviewed scientific papers, such as Nature Reviews Microbiology, GCA, and EST, and so on. His research has a high index of 48. In the year of 2002, Professor Shi joined the Geomicrobiology Group of Pacific Northwest National Laboratory, USA, as a grade 3 research scientist and PI. He was then promoted to grade 4 and 5 research scientist in the year of 2006 and 2012, respectively. Right now, Professor Shi moved to CUG Wuhan in 2016. His research have focused on the molecular mechanisms of microbiome, EET, and the biotechnology application of the microorganism with EET capability, such as bioremediation, microbial fuel cells, and electrobiosynthesis. In the year of 2020 and 2021, Professor Shi was invited to nominate Nobel Prize in Chemistry by the Royal Swedish Academy of Science. Right now, let's welcome Professor Shi for his presentation.
Thank you, Professor Gao, and good morning, everyone. And as Professor Gao mentioned, I'm at the China University of Geosciences, short for CUG, in Wuhan, not in Beijing. I've been in the CUG Wuhan for nearly 7 years. Prior to that, I had worked at the Pacific Northwest National Laboratory, or PNNL, for 14 years. Therefore, today, I will report my previous work at PNNL very briefly. Then I'm going to report to you to our current work at the CUG Wuhan in more detail. PNNL is at the southeast corner of Washington State. So here's the main campus of the PNNL. Immediately north of the main campus of PNNL is the Hanford site. So the Hanford site is very large. It's 586 square miles, and that site is highly polluted with different contaminants, including the radioactive contaminant. So for this reason, the US Department of Energy established the Environmental Molecular Sciences Laboratory, or EMSO, inside the main campus of PNNL about 20 years ago. So the EMSO launched the EMSO Biogeochemistry Grand Challenge project, whose major goal is to reveal the molecular mechanism for electron transfer between microorganisms and iron-containing mineral. The reason they focus on this interaction between microorganisms and iron-containing mineral is that the iron-containing mineral are abundant in the soil and subsurface sediments, where they electrically support microbial growth in at least 4 different ways. First, so after iron-3 reducing microorganisms oxidize organic matters, they can transfer electrons to the mineral containing iron-3 for anaerobic respiration. And in contrast, the iron-2 oxidizing microorganisms can use structural surface iron-2 of the mineral to reduce oxygen, CO2, and nitrate for growth. We know some of those minerals are semiconductives. So this mineral can transfer electrons from one species of microorganism to another species of microorganisms. And finally, these iron-containing mineral containing both iron-2 and iron-3. So under the one condition, they can receive electrons from iron-3 reducing microorganisms. Then when the condition change, they can donate electrons to the iron-2 oxidizing microorganisms. Most importantly, the electrical interplay between the microorganisms and iron-bearing minerals impact greatly on the mineral formation and the weathering. So in this figure, the iron-3 containing mineral hematite is in red, and the iron-3 reducing bacterial Schumanninaodanesis MR1 are in green. To exchange electrons with these extracellular minerals, Schumannina and other iron-3 reducing microorganisms And iron-2-oxide microorganisms, they evolve this what we call extracellular electron transfer pathway, short for EETP. This EETP coupled intracellular metabolic activity to the extracellular redox transformation of ion. I'd like to point out this EETP differ from the electron transfer chain, or usually called ETC, in at least 3 different ways. First, EETP transfer electrons across cell envelope, while ETC transfer electrons only in the cytoplastic membrane. And ETC, the purpose is to generate this proton gradient for ATP synthesis, while EETP is not. And finally, EETP can transfer electron both inside out as well as outside in, where ETC can only transfer electron in one direction, from complex 1 to complex 4. So my work at the PNNL is identify and characterize this extracellular electron transfer pathway. So in 2016, when I left the PNNL, these 4 bacterial electron transfer pathway were well characterized. Among them, this mido-reducing or MTR pathway of Schwann adenosine MR1 is the most vigorously investigated and best characterized. So in this pathway, the Simon A in the cytoplasmic membrane oxidize quinol and then transfer release the electrons to the paraplasmic FCC3 and STC. And these 2 proteins transfer electrons across the paraplasm to the MTR ABC and out membrane. And MTR ABC transfer electrons from the paraplasmic space across the out membrane to the exterior of the cells. And outside cells, MTRC then transfer electron directly to the mineral surface. Similarly, Geobacter sulfur reducers use this PCC pathway, transfer electrons from cytoplastic membrane across cell envelope, then to the mineral surface. Well, these rhodopseudomonas plastrix TIE1 and stereoxidants lithotrophicus ES1 use this PIO pathway and MTO pathway respectively, oxidize ion 2 extracellularly, then transfer this release electron across the cell envelope all the way to the quinol in the cytoplastic membrane. So I'd like to point out that these PCC proteins are different from these MTR proteins. However, MTR protein, proteins, and MTO proteins are homologous to each other. And the Diane Newman's group at Caltech identify and characterize this PIO pathway. We at the PNNL, as well as our collaborators, identify and characterize these 3 pathways. Many people are involved in this work. However, these pictures show some of the people involved here. And John Zakra and Jim Fredrickson, who co-led the AMSO biogeochemistry Grand Challenge project, as well as subsequent PNNL SBR SFA project. Very sadly, John passed away 2 years ago. Kevin Russell and I co-led the molecular part of this project. David Richardson, Tom Clark, Kay White, and Naroko Baden, as well as the Marcus Edward, they are from the University of East Anglia at the United Kingdom. So they did the most work of characterize these proteins, including the molecular structure determination of MTR-ABC complex, as well as other proteins. Zizi was a postdoc of the John Zakra, and Eric Roden of the University of Wisconsin-Madison was our collaborator. And the interactions between the microorganisms and iron-containing mineral are involved in the biogeochemical cycling of not only iron, but also the carbon, nitrogen, sulfur, arsenic, and iodine. And this interaction impact the feed and transport of a variety of environmental contaminants, such as arsenic, iodine, cadmium, technetium, and uranium. And finally, this interaction causes the acid mine drainage formations. And the microorganisms with these extracellular electron transfer capabilities have been used in a variety of the biotechnological applications. First, these microorganisms can be used for the bioradiation of the environmental contaminant, such as the radioactive uraniums. They have been used for biomining of copper and gold, and the picture show is one of the biomining factories found in the Chile. Actually, this is probably the largest microbial factories. This microorganism has also been successfully used for the microbial fuel cells. This is just like generators, so they can produce electricity. And this is the one is shown was developed by U.S. Navy for the generating electricity and by bacteria. And some of these microorganisms can produce these nanoparticles, at room temperature. And this nanoparticle has a unique application, such as killed the tumor cells. And most importantly, I believe, is this microorganism can also use electricity from the renewable sources to fix CO2 and produce these other chemicals, such as advanced biofuel in the carbon neutral way. And finally, the shield and the geobacter can also cause the corrosion of carbon ion, which is iron 0. So the corrosion of iron 0 is iron 0 oxidized to the iron 2. So investigation the molecular mechanisms of this extracellular electron transfer will probably find a way to treatment this to prevent the iron 0 corrosions. So after I moved to the COG Wuhan about 7 years ago, we also discovered some new mechanisms, so microbial extracellular electron transfer. The first one is the extracellular electron transfer pathway of the Bacillus ferrooxidants for iron 2 oxidation. So Bacillus bacteria, not the bacillus. The ferrooxidants was first isolated from the acid mine drainage site in the US. It is the first microorganism demonstrate conclusively they can eat or oxidize iron 2 for the chemolithotrophic growth at a pH of 2. Because they can fix CO2 and nitrogen, so it is one of the crucial players in producing the organic carbon and the nitrogen in the environment that is acidic and poor in the nutrients such as AMD side. So we know the AMD is a very environmental issue worldwide, and which occurs when the mineral containing sulfide, such as pyrite, interact with water and oxygen, and they abiotically produce iron 2 sulfate and protons. At the acidic condition, in the absence of any microorganisms, this iron 2 oxidized to iron 3 is very slow. This is the rate limiting step for the AMD formation. So the ferrooxidants and other iron 2 oxidizing microorganisms can increase this step by the order of several order of magnitude. So it's one of the key AMD causing microorganisms, and is often found in the AMD site worldwide. And this microorganism can be also used for the biomining of the copper and the gold, as I showed in the previous slides. So it is also important of industrial microorganisms. So previously, it is shown that the microorganism oxidize iron-2 extracellularly, and then they can transfer electrons inward across cell envelope to the cytoplasmic membrane, where it can either reduce oxygen or the NAD. So these proteins are involved in these pathways. And my colleague, Professor Peng Zhaofeng, and his graduate student Liu Ziyu focused on the interaction of these three proteins, where the in vivo chemical cross-linking method under the iron-2 oxidizing condition. So they use two different kinds of cross-linker. First one is called DSS. This DSS is hydrophobic, so they can penetrate into the cells. The results show that these chemicals cross-link these two proteins, CYC2 and IOS, as well as IOS and CYC1. However, its analog DS3 is water soluble, so they cannot cross the outer membrane. And so it can only cross cross-linking these two proteins. And then that indicate IOS interact with CYC2 very close to the cell surface. So based on these results, we propose that after the iron-2 oxidation, the CYC2 transfer electrons to the IOS inside, which is inserted CYC2, and IOS then transfer electrons across the outer membrane to the IOS in the paraplasmic space, and then the IOS transfer electrons to the CYC1. The second mechanism is extracellular electron transfer pathway of sugar nanoadenosine MR1 for iodate reduction. So iodine, as you know, is an essential trace element for the human and other vertebrate because it's the key component of human thyroid hormone, and also for the animals. So imbalance of this iodine inside the humans can cause a variety of these human disease, such as goiters. So in China and also other countries, high iodine groundwater exist in these countries, so which affect the health of many peoples. In addition, the iodine is also the trace element of the environmental significance. For example, this radioiodine, iodine 129, whose half-life is 16 million years, is a major risk driver in the herbicide that I mentioned before. And also global biogeochemical cycling of iodine impacts air quality and climate. So my graduate student Guo Jinzhi found that DMS-EFAB of sugar nanoadenosine MR1 reduce iodate extracellularly to the hypoiodous acid. Then MTR-ABC, which reduce and as well as they produce HIO and hydrogen peroxide. So MTR-ABC then further reduce hydrogen peroxide to facilitate DMS-EFAB mediate iodate reduction. So the MTR pathway I mentioned in the beginning, and also transfer electrons from the cytoplasmic membrane to the DMS-EFAB and MTR-ABC on the bacterial surface. So how the HIO converted to the iodide currently is unknown. One is abiotically to convert to the iodide and iodate, or biotically to reduce to iodide and oxygen. Currently, we are investigating which this microorganism is described in the MR1. So Guo Jinzhi's results is also found that with the microorganisms with the capability for the extracellular reduction of iodine are isolated from a variety of environmental habitats, such as the lake, rivers, and the subsurface rock in the East and Southeast Asian, North American, Africa, and North American. And most importantly, these microorganisms are found in the marine environment, such as Arctic Ocean and Antarctic coastline, marine coastline, as well as Atlantic Ocean, Indian Ocean, and the Pacific Oceans. So widespread distribution of these bacteria with capability for extracellular reduction of iodate around the world suggests the significant importance of these microorganisms in the biogeochemical cycling of iodines. As we mentioned before, the high-level groundwater iodine is also a major issue in China, especially the North China Plain. So my colleague, Professor Jiang Zhou, and our graduate Huang Minghui found that the iodine level in the groundwater in the North China Plain vary significantly. Some only have 2 microgram per liter, and some has more than 1,000 microgram per liter. So what they did a comparative analysis of this groundwater to see what kind of microorganisms in this water, and the results of their results are very interesting. What they found is the iodate reducing microorganism, iron-3 reducing microorganisms, which we mentioned before, as well as the sulfur reducing microorganisms, all contribute to iodide enrichment. I forgot to mention the major species of this in this groundwater is iodide. So this is how we believe why the forms is very high iodide concentration in this groundwater in the northern China Plain. So in addition to the iron, as well as the iodide and other non-living things, these microorganisms can also transfer electrons to other microorganisms. So this phenomenon called direct interspecies electron transfer, or DIET. So the first DIET was demonstrated from the co-culture of geobacter metal-reducens and with geobacter sulfur-reducens. So in this co-culture, the metal-reducens oxidize ethanol, okay, then they transfer electrons directly to the sulfur-reducens, and the sulfur-reducens use these electrons to reduce the fumarate to the succinate, okay. These nanowires play a critical role in this direct electron transfers. Right now, the known nanowires of geobacter, including the POA and the multi-heme C-type cytochrome OMCS, OMC, and OMCZ, and OMCT is homolog of OMCS. So my graduate student, Luo Yin, and she found the POANs of both metal reducens and the sulfur reducens, and they play a critical role in the co-culture, and in which the POAN of the metal reducens is more important than that of the geobacter sulfur reducens in the direct electron transfer from the metal reducens to the sulfur reducens. So Professor Peng and our graduate student Jiang Jie, He Peng Chen, and Luo Yin found that the role of these nanowires of the sulfur reducens in the co-culture vary substantially. Okay, For instance, the role of the OMC in the co-culture is minor, and OMCZ like the PRAN, which are indispensable because if you knock this gene out, there's no co-culture form between these two bacteria. For the OMCS and OMCT, which is interesting, you knock them out just by one genes, they reduce the co-culture, but they can still form the co-culture. However, if we knock out both OMCS and OMCT, and then there's no co-culture form. So these two proteins have overlapping roles, and both together, they are also indispensable for the direct electron transfer from the metal reducings to the sulfur reducings. And Professor Peng and his graduate student Wang Yinghui also found that the pure AC and OMCS of the geobacter sulfur reducers, they can interact with each other extracellularly, where direct contact during the co-culture. So these we also did the research on the biotechnological applications of Shield nano-adenosine 1 and geobacter species. So the first work is use the Fenton reaction driven by Shield nano-adenosine 1 to degrade this organic contaminant, and these are what work of my graduate student Smei, who graduated about 2 years ago. So Shield nano-MR1 is very interesting microorganism. So under the anoxic condition, they can reduce ion 3 to ion 2, as I mentioned. However, under the oxide condition, though, they can use oxygen to produce hydrogen peroxide. So ion 2 and hydrogen peroxide can react abioctally through the Fenton reaction to produce hydroxyl radicals. These hydroxyl radicals can oxidatively degrade many organic contaminants, such as the BDE47. So her results also found that these hydroxyl radicals oxidize degrade the BDE3, which only has one bromine atom, is much faster than degrade this the BDE has 10 bromine atoms. So, and her second discovery was that these ion-containing minerals, they facilitate the degradation by increasing the hydroxyl radical production. So the advantage of this method is that you don't have to add the hydrogen peroxide. Okay, the second one is the ion is recycled. So in this way, it can reduce the cost greatly. And it also represents an environmental friendly way to mediate organic contamination. So we also interested in to increase electric output with synthetic biology approach. So these results of my colleague, Professor Jiang Yongguang, and his graduate student, Wang Zigao. What they did is the overexpression of these nanovirus I have mentioned before, and they can find the overexpression of these proteins. They can increase the biofilm formation, as well as also increase the electrical current production. So they can generate more electricity. So the goal of this research project is combine the integrated to develop an integrated device that combine the microbial fuel cells. Microbial fuel cells oxidize the organic waste, then they harvest the electricity, then they transfer electricity to the cathode. So then this another microorganism, they can use the electrons to fix this CO2 to produce the material we want. So the another part of this project is to electrical CO2 fixation, also where the synthetic biology approach. So the method, the way we fix CO2 is use this reverse TCA cycle. We know the TCA cycle is very common among the many microorganisms. So reverse TCA cycle is a way to fix CO2. And previously, Derek Lively's group in the US, they used synthetic biology group to create the new strains and to fix CO2 with the electrical electrons and where these TCA cycles. So inspired by his by the Derek Lively team's group's work, my graduate student Luo Zihao, and then he found that the geobacter sulfur reducents not fix CO2 electrically, not only fix CO2 electrically very well, and also he further converts the CO2 to the ethanol, which is a biofuel at the rate of 49-53%. So I believe this microbial CO2 fixation with the renewable electrical sources is a carbon neutral way to produce biochemicals, and is represent new frontier of electro-microbiological research. So another frontier of the research related to the microbial extracellular electron transfer is the photosynthesis by the non-photosynthetic microorganisms. So in this way, this nano-sized semiconductors, which labeled in yellow, which can be made from the minerals, which are very, very cheap in the natures, and they can produce photoelectron when there is a light available on the microbial surface. Then this photoelectron can be transferred into the inside the microorganisms, and these microorganisms then use the electrons to fix CO2. So photosynthesis with the semiconductors and the non-photosynthetic microorganism open a new way for the solar to the chemical energy conversion. I like to point out this is still in the infancy. We're still in the proof of principle stage. So finally, I'd like to thank my current and former colleague at the COG Wuhan and PNNL, as well as my current collaborators and the former collaborators for the this long, very, very long history of collaboration. The results I presented here nearly all through the collaboration with this group of highly intelligent and very productive researchers. And I also like to thank the agency who funded my research for the last over 20 years. So thank you for listening. I'm ready to answer your question. So I was told it's about 40 minutes talk and about 20 minutes question and answer. Thank you. Ready for your questions.
Thank you, Professor Shi. Now it's the question time. Any people who have a question and maybe some good advice and some topic want to discuss with Professor Shi right now is a good time. Welcome. Okay. I have a question. It's in fact, it's not really a question, but I was very interested in that how to use this technology to deal with AMD in the field set, Professor Shi.
Well, for the AMD treatment, how to remediate AMD, which is not very easy. And I have to tell you, I'm not expert in the bioremediation of AMD. I think Professor Su Wensheng, and he's in the Guangzhou, and he's very good at it. In this case, we are more interested in is the mechanisms, okay, how they transfer electron, both inside out, from outside in. And my only experience is that you have to find a way to reduce its acidity. It's not by these microorganisms, and you have to use some alkaline producing microorganisms to balance the pH, because the AMD is the most damaging thing is extremely low pH. So I have to tell you, I have no very good way to give the suggestions how to use this microorganism to treat to remediate the AMD problem. But this microorganism can be used for the biomining, so it's industrial microorganisms. And I would say the probably way to increase the efficiency, to degrade these the metals, and to increase the copper, and then to how to recover it. Probably we can think about that, but this is one not for the environmental applications. I hope I answer your questions.
Thank you. It's very, very suggested for me. And another question is, you have done some very frontier work, and could you mind to give the young research some suggestion to how to focus on the like international topic and to do their research work?
Okay, well, this the I'm not sure my my advice will be useful to everyone. I always believe that the other people's success, you can always to learn from it, but you cannot copy it. So that's my first suggestion. So I have to tell you, I'm not trained as a geomicrobiologist or the environmental microbiologist. So I was trained to study human disease. And about 20 years ago, I was went to the P9 alpha interview, and everyone talking about Schwannana. That's the first time I heard Schwannana. I never heard of Schwannana before. So the thing is, they asked me, because I have the background of the protein chemistry, they asked me to do the protein purification for the MSO biochemistry grand challenge, and whose major goal is to do the three-dimensional structure of these proteins. I think that's very, very, I should say it's very far. This goal wasn't set by me, but it's a really excellent goal. Okay, at that time, people studied geomicrobiology as well as environmental microbiology. They didn't pay attention, especially in the geomicrobiology, they didn't pay attention to the mechanisms, okay? And they are just fascinated about how these bacteria can transfer electron outside of the cells. Okay, they can do the genetic study. So my expertise for this project is because I know how to do the protein purification. So I purified the protein and then I gave them to the UEA groups in the UK. So they did the three-dimensional structure. That's a pioneer work, okay? So in the beginning, when we did that, and many people wasn't very interested. They said that's not necessary. It's not very, at that time, people not very supportive of this work. However, after we published work, we showed how these things work, the electron transfer across the outer membrane, and people began to realize this is a very important area. So as I showed the the results. It's not our results. Let me see. I can go back. For some reason, I cannot go back. So the nanowires, okay, and for the last 4 years, there is a very good publication in the Nature Cells and all those publications of the nanowire structures. Okay, I think this is opened a new way how this the nanowire, what it looks like, how the electron can transfer very long distance. So I'm not saying we laid the foundation for the nanowire structure determination. At least we showed the way how, if you want to know the fundamental of this extracellular electron transfer, one major point is the structure determination of these proteins. So that's the reason we did the pioneer work of this extracellular electron transfer. We're not just doing the traditionally culture and gene knockout. We go to the protein, we look at the structure, and then we did many work on the proteins characterization and related characterization data with the other observations. So this is I think that's this very new way to do this geomicrobiology group. Nowadays, you can see that the people can also follow that the path and do the molecular structure of these very important and very interesting proteins, which if you do not do this work, then you never know how this electron can be transferred for a very long distance. I'm not sure I answered that your questions. This is the one I can give to the new researchers. You have to pick up the very important issue, and you have to be patient. Okay, I'm very just lucky. The person who pick up this important issue is not me. They pick up this issue. They hire me to carry out this work, and then I'm very lucky for the U.S. Department of Energy support this kind of work for about 10 years. So 10 years you have the support, and then we have these collaborators. They are very smart, much smarter than me. So now we made the progress in this area. That's the I'm just lucky, and also you have to be to have the ability to get the job done. That's my answer.
Thank you, Professor. I think your work will be well supported by a Chinese government.
Oh yeah, you can see how many funds I got for the last 7 years, and also from CUG Wuhan. So.
And there is a question from someone. He asked that is it possible for the electron to move from the underground environment to the surface, by the nanowires?
Right now, the nanowires, the longest is about centimeters long. That's not by the nanowires, that's by the cable bacteria. Okay, the cable bacteria, they can transfer electrons about at least centimeters. What they did is in the under the condition without the oxygen, so they oxidize the hydrogen sulfide, then they transfer electrons through the layer of the mud and to the surface, so they have oxygen. Okay, so that's called the cable bacteria. And the cable bacteria, we are doing this kind of work, but we haven't had any publications. But the best the group to study the cable bacteria is in the Netherlands, so they are very good at it. And if you want, you can look at their publication. But remember the bacteria, microorganisms, they are very tiny. Okay. The micron scale. So they move the electrons not very far away. Like if you're doing those field scales, and this is not the electron moving for them. This is very the micro scale and the nano scale we are talking about. It's not talking about the field scale. That's my answer.
Okay, thank you, Professor Shi. Limited by the time, I think our talk is going to close. And here we want to thank Professor Shi again for his wonderful presentation. And this talk is over. Thank you, Professor Shi.
Okay, thank you. And thank you very much. Bye.
Thank you, thank you.
Bye.
Bye.
Thank you, Mr. Shi. I wish Mr. 我希望我在说中文吧，我希望那个石老师下次有机会给我们再做一次报告，因为这个报告虽然不是我的专业，但是我觉得非常有前景。
谢谢谢谢，谢谢您。我这个我这个可能我这个电脑可能出了事，
没关系，
死机了，看不上去。然后那个行，你要让我做报告，我肯定去了。那个就是江院士的主持的那个期刊，我觉得这个期刊那个角度很高，而且他也抓到了那个跟上了那个时代的命脉，对不对？然后是大家都感兴趣，因为我们做环境嘛，最后还是要跟那个健康有关。对，我去年去参加那个全国环化大会，那个江院士也是专门讲这个期刊，我觉得这个期刊很好的。然后那个希望他把它变成一个国际一流期刊，我觉得一定能变成。
谢谢石老师，希望你还积极给我们投稿。
我们要有稿件，一定要高质量的稿件，一定是发到你那儿去。
谢谢石老师。
那我们先向石老师约稿了。
约稿，正式约稿。谢谢石老师。
OK，拜。
好，也谢谢高老师这精彩主持。好的，谢谢。下次再见。
好，再见。